Extranuclear inheritance or cytoplasmic inheritance is a transmission of genes that occur outside the nucleus. It is found in most eukaryotes and is commonly known to occur in cytoplasmic organelles such as mitochondria and chloroplasts or from cellular parasites like viruses or bacteria. Mitochondria are organelles which function to transform energy as a result of cellular respiration. Chloroplasts are organelles which function to produce sugars via photosynthesis. In plants and algae, the genes located in mitochondria and chloroplasts are very important for proper cellular function, yet the genomes replicate independently of the DNA located in the nucleus, which is typically arranged in chromosomes that only replicate one time preceding cellular division. The extranuclear genomes of mitochondria and chloroplasts, however, replicate independently of cell division. They replicate in response to a cell's increasing energy needs which adjust during the cell's lifespan. Since they replicate independently, genomic recombination of these genomes is rarely found in offspring contrary to nuclear genomes, in which recombination is common. Mitochondrial diseases are received from the mother. Fathers don't, as sperm do not contribute, but a sperm contains a mitochondrion for its energy production. Extranuclear transmission of viral genomes and symbiotic bacteria is also possible. An example of viral genome transmission is perinatal transmission. This occurs from mother to fetus during the prenatal period, which begins before birth and ends about 1 month after birth. During this time, viral material may be passed from mother to child in the bloodstream or breast milk. This is of particular concern with mothers carrying HIV or hepatitis C viruses. Examples of cytoplasmic bacteria have also been found to be inherited in organisms such as insects and protists. Mitochondrial inheritance is the study of how mitochondrial genes are inherited. Mitochondria are cellular organelles that contain their own DNA and RNA, allowing them to grow and replicate independent of the cell. Each cell has 10,000 mitochondria, each containing 2 to 10 copies of its genome. Because mitochondria are organelles that contain their own genome, they follow an inheritance pattern different from simple Mendelian inheritance, known as extranuclear or cytoplasmic inheritance. Although they possess their own genetic material, mitochondria are semi-autonomous organelles because the nuclear genome of cells still codes for some components of mitochondria. Mitochondria are able to divide independently without the aid of the cell. The chromosomes of mitochondria are replicated continuously by the enzyme DNA polymerase, which is a strand of DNA having different points of origin. Initially, one of the parental strands of DNA is displaced while the other parental strand is being replicated. When the copying of the first strand of DNA is complete, the second strand is replicated in the opposite direction. Mutation rates of mitochondria are much greater than that of nuclear DNA, allowing mitochondria to evolve more rapidly than nuclear genes. The resulting phenotype, cell death, inability to generate energy, or sign of mutation that has no phenotypic effect, is dependent on the number and severity of mutations within tissues. During fertilization, mitochondria within the sperm are excluded from the zygote, resulting in mitochondria that come only from the egg. Thus, mitochondrial DNA is inherited through the maternal lineage exclusively without any recombination of genetic material. Therefore, any trait coded for by mitochondrial genes will be inherited from mother to all of her offspring. From an evolutionary standpoint, mitochondrial Eve genes were inherited 200,000 years ago. Other women living at the time did not succeed in passing on their mitochondria because their offspring were only male, although the living descendants of those other females were able to pass on their nuclear genes. Only mitochondrial Eve succeeded in passing on her mitochondrial genes to humans alive today.
Plasmid is a small DNA molecule that is physically separate from and can replicate independently of chromosomal DNA. Within a cell, most commonly found as small circular. A cell most commonly found as a small circular double-stranded DNA molecules in bacteria. Plasmids are sometimes present in archaea and eukaryotic organisms. In nature, plasmids carry genes that may benefit survival of organisms, for example, antibiotic resistance, and can frequently be transmitted from one bacterium to another, even another species, by horizontal gene transfer. Artificial plasmids are widely used as vectors, the molecular cloning serving to drive the replication of recombinant DNA sequence. Within the host organism, plasmid sizes vary from 1 to 1000 kbp. The number identical plasmid in a single cell can range anywhere from 1 to 1000 under some circumstances. Plasmids can be considered part of the mobilome because they are often associated with conjugation, a mechanism of horizontal gene transfer. The term plasmid was first introduced by American molecular biologist Joshua Lederberg in 1952. Plasmids are considered replicons, capable of replicating autonomously within the suitable host. Plasmids can be found in three major domains: archaea, bacteria, and eukarya. Similar to the viruses, plasmids are not considered by some to be form of life. Plasmids are naked DNA.
Today, I will going to talk about the criteria of extrachromosomal inheritance. This will be focusing on the characteristic and special features of extrachromosomal inheritance, or also known as cytoplasmic inheritance. So, the first criteria is the reciprocal difference. Characters which are governed by cytoplasmic inheritance invariably exhibit marked differences in reciprocal crosses in F1, whereas in case of nuclear inheritance, such differences are not observed except in case of sex-linked genes. So here, this is the diagram or example of a Mendelian inheritance, and this is the cytoplasmic inheritance. So here, the female having the allele of big Y and small y is crossed with the male having an allele of big R and small r. So the resulting offspring of them is this. And if you're going to reciprocate them, the female will be having the allele of big R and small r, and the male will be having the big Y and small y. If we are going to cross them, the resulting offspring of them will be the same as above. But in cytoplasmic inheritance, let's say the female is color green and the male is color white. If we're going to cross them, all of their offspring will be color green. And if you're going to introduce the reciprocal crosses, the female will be going white and the male will be going green, and all of their offspring will be going white. Next of it is the maternal effects. In case of cytoplasmic inheritance, distinct maternal effects are observed. This is mainly due to more contribution of cytoplasm to the zygote by female parent than the male parent. Generally, ovum contributes more cytoplasm to the zygote than sperm. So let's talk about the fertilization. So here in the fusion of the egg cell and the sperm cell, only the nucleus of the sperm cell are going to introduce to the egg cell. Here is the zygote, where the nucleus of both sperm and egg are fused together. All the cytoplasmic material inside the zygote is the same with the egg cell before fusion. So we can say that
all of the organelles and cytoplasm in the zygote is the same with the egg cell which came from the mother. So next is the mappability. Nuclear genes can be easily mapped on chromosomes, but it is very difficult to map cytoplasmic genes or prepare linkage map for such genes. So in the Mendelian inheritance, it is easy to map the genes, while in cytoplasmic inheritance, it is difficult to map the genes. So next is the location of the genes. The genes in the Mendelian cross is usually found on the chromosome, while in the cytoplasmic inheritance, the genes is mostly found in the chloroplast and in the mitochondria. So last criteria for this is the governed by plasmid genes. True cases of cytoplasmic inheritance are governed by chloroplast or mitochondrial DNA. In other words, plasmid genes are made of chloroplast DNA and mitochondrial DNA. According to endosymbiont theory, mitochondria and chloroplasts were once free-living organisms that were each taken up by a eukaryotic cell. Over time, mitochondria and chloroplasts formed a symbiotic relationship with their eukaryotic host. Although the transfer of a number of genes from this organelle to the nucleus prevents them from living independently, each still possesses genetic material in the form of double-stranded DNA.